Imagine sitting with a loved one who's struggling with depression or a different psychiatric condition. Years later, you watch symptoms like cognitive and memory impairment emerge. They are diagnosed with dementia, and now you wonder, were the signs there all along, but presenting as something different at an earlier stage? Thanks to the remarkable work of researchers and medical professionals, the average lifespan of an individual has increased significantly. However, a longer lifespan is often accompanied with dementia-related conditions, or neurodegenerative diseases. Neurodegenerative diseases are characterized by the progressive decline in cognitive and motor function, primarily impacting the elderly population. However, we are still working to better understand these conditions as well as develop effective treatments. This brings forward a question in my research: could we better predict neurodegeneration prior to the manifestation of symptoms? Every day, millions of individuals are treated for psychiatric conditions such as anxiety, depression, mood disorders, but our care often stops at managing these symptoms in the moment. What if these symptoms were sometimes the earliest signs of something deeper, such as the earliest stage of neurodegeneration? We now know that psychiatric conditions are the most common co-occurring condition and often precede the onset of neurodegeneration. What we still don't know is how or why this happens. Not everyone with dementia has depression, and not everyone with depression will go and develop dementia. Somewhere in that uncertainty lies a biological gap, a missing link in our understanding of who is most at risk and why. My research aims to bridge this gap using a gene that can help us reveal how both of these conditions unfold, the FMR1 gene. Changes in this gene can increase the risk of developing brain-related conditions across different life stages, including depression and neurodegeneration. It provides us with the window to examine the relationship between psychiatric and neurodegenerative phenotypes, which has been difficult to untangle due to the late manifestation of symptoms that we see in neurodegeneration. My research uses post-mortem brain tissue from individuals with changes in this gene, as well as a psychiatric or neurodegenerative diagnoses. Using this tissue, I will uncover cellular and molecular mechanisms that can help us reveal the relationship that may link psychiatric with neurodegenerative symptoms, and help us as well. It'll also help us predict neurodegeneration before it begins, develop effective treatments for both conditions, as well as raise awareness that psychiatric symptoms are not only an issue for the present. Thank you.